The genetic disease that we chose to research for our final project is breast cancer. Breast cancer can occur in both men and women, as it is a cancer formed within the cells of the breasts. Breast cancer may be inherited due to a combination of environmental and genetic factors. Studies indicate that hereditary genetic alterations account for 5 to 10% of occurrences of breast cancer, with environmental variables and random genetic mutations accounting for the other 90 to 95% of cases. Gene mutations that cause abnormalities in specific genes, such as the BRCA1 or BRCA2 genes, significantly raise the risk of breast cancer. These genes play a role in both preserving genomic stability and mending damaged DNA. A higher risk of developing breast cancer, as well as ovarian cancer and other cancers, can result from mutations in these genes. Proteins involved in DNA repair pathways, BRCA1 and BRCA2, are among the altered proteins frequently seen in individuals with hereditary breast cancer. These proteins' capacity to repair damaged DNA is compromised by mutations, which increases the risk of developing cancer and causes genomic instability. Furthermore, proteins including TP53, PTEN, and PALB2, which are linked to additional hereditary breast cancer syndromes, are also essential for DNA repair and cell cycle regulation.
The popular 23andMe test examines only 44 out of over 4,000 variants in the BRCA1 and BRCA2 genes, which are the most important genes they analyze for variants linked to breast cancer. These genes provide instructions for making a protein in the cells of the mammary gland that acts as a tumor suppressor, preventing unnecessary cell growth. Since these mutations impact the structure or expression of this protein in the mammary gland, it makes sense that it leads to an increased risk for breast cancer. A specific variation tested by 23andMe is a deletion on chromosome 17 that changes protein function. If you have a harmful BRCA mutation, such as the one previously mentioned, you have a likelihood of 45 to 85% for developing breast cancer in your lifetime, compared to 12% for the general population. Once again, 23andMe only tests for 44 out of more than 4,000 variants in the BRCA1 and BRCA2 genes, so there are many variations that impact risk but are not included in their test. They also only test for variants in 2 out of the 26 genes known to be associated with breast cancer, so they do not perform a comprehensive test.
Our SNP chip test consists of both the 14 BRCA1 and BRCA2 SNP variations tested by 23andMe, as well as over 10 more SNP variations in the CHEK2, TP55, and ATM genes, which have all been linked to breast cancer. The ATM gene encodes for the ATM kinase protein responsible for the detection and repair of damaged DNA strands. The SNP that we will include on our test and believed to be the best predictor for the risk of breast cancer is the RS289-04921 variation. This missense variation is a SNP found on the ATM gene, which was previously mentioned. The two possible genotypes for this variation are homozygous for the T allele and heterozygous for the T and G alleles, which comes with the increased risk of breast cancer. Despite the ATM gene being expressed in all types of tissues in the body, the RS289-04921 SNP has not been linked to any specific change in the gene's function. Regardless, those who have the variation have shown to have a 240% increase risk of breast cancer from a base risk of 1.51%.
Genetic variants associated with breast cancer, such as those in BRCA1, BRCA2, PALB, ATM, and CHEK2, explain only a portion of the excess familial risk. Many other common genetic variants identified through genetic testing may have low penetrance and only cause a slight increase in risk. For example, the allele RS2056116 plays a role in the risk for breast cancer. However, this is only if the allele is an AG or GG pair, which only makes one's risk as low as 18% or as high as 40%. Genetic variants, considering that there are over 4,000 variants of BRCA1 or 2, the amount of possible allele combinations increases drastically as well. Individuals may overestimate their risk based solely on genetic testing results. While some variants have been identified, many others may still be unknown or not fully characterized. Additionally, environmental factors such as alcohol consumption and physical activity level are also involved in assessing risk of breast cancer. However, this cannot be determined via a genetic test. Furthermore, understanding the baseline risk of genetic cancer in the general population is essential. The heritability of breast cancer is estimated to be around 30%. Even if a person tests positive for a specific genetic variant associated with breast cancer, the actual increase in their risk may be relatively small. These reasons will be considered and will be made transparent to the public for our test.